Hi, I'm Jonathan Brescher, and I'm going to show you how to use the shared clustering application as a way to get a lot more information from your Ancestry DNA matches. First, let's get some software. The simplest way to find the shared clustering application is to go to Google and type shared clustering download. The first link on the page will take you straight to the download. You can then click on the download link and launch the installer, which I did earlier. Before leaving this page, you should notice that there are many more links further down. These links have detailed documentation and examples of using shared clustering to help with your DNA research. The next step is to get your data from Ancestry. Shared clustering runs entirely on your own computer, so nobody knows your password and nobody has access to your data but you and Ancestry. In the Download tab, enter your username and password and click the Sign In button. I'm going to start the download first and then describe the rest of the screen. So I'll click on the Get DNA Matches button and accept the default file name. There are different amounts of data that you can download. For this demo, we'll just download the matches that Ancestry calls fourth cousin and closer. This is the fast and incomplete option in the middle of the screen here. It will download the matches over 20 centimorgans. That may be enough data for many people, especially for adoptees who are looking for their closest relatives, parents and grandparents. If you are doing research into more distant relatives, you normally will want to access all of your data down to Ancestry's lowest limit of 6 centimorgans. This is the slow and complete option. Most people have never looked at any of their matches under 20 centimorgans, except for maybe the few that showed up in through-lines. There's a lot of data in the matches below that cutoff, just waiting for someone to look at it. When the download is complete, you can click the button to continue working in the cluster tab. In this demo, we'll start off by looking only at the closest matches, the ones over 90 centimorgans. These are the matches that Ancestry calls third cousin and closer. So we select that option in the middle of the screen, click the Process Save Data button, and your clusters will open automatically in Excel. We can zoom out to get a little better view. What exactly are we looking at? First, remember that we limited these clusters only to the closest matches, the ones over 90 centimorgans. That limit means that each match is probably related to you through one of your grandparents. Some of your closest matches, your aunts and uncles and first cousins, are related to you through two of your grandparents. The cluster diagram shows these relationships visually. There are several ways to read the diagram. Each row represents one of your matches. Each column represents a possible shared match to the match in the row. If the shared matches of the person in the row include the person in the column, then the intersecting cell is shown in color. Large square areas of color represent groups or clusters of people who mostly all match each other. In this example, a quick look shows that there are 4 squares. 1, 2, 3, 4, where the first 2 squares overlap and the last 2 squares overlap. Each square represents matches who are related to this test taker through one of their grandparents. The overlapping areas represent closer relatives who match the test taker through 2 grandparents. Simple. These relationships are also shown numerically on the left side of the diagram, along with several other bits of data from the Ancestry site. The shared centimorgans and shared segments columns are especially interesting. You can see that the matches in the overlap area here really do have higher shared centimorgans than the matches on either side. This diagram shows a common situation. From the shared centimorgans column, you can see that the matches in the overlap area have shared centimorgans in the range of 400 to 800 centimorgans. These are likely first cousins to the test taker. The matches on either side of the overlap area have shared centimorgan values in the range of 90 to 200 centimorgans. These are second cousins to the test taker. The overlap shows visually that the first cousins are related to both sets of second cousins, but the two sets of second cousins are not related to each other. With very little effort, this test taker now has isolated their matches related to each parent and to each of his 4 grandparents. Clustering doesn't tell you which cluster goes with which grandparent. You need to research that part, but often the research is easy. In this case, the test taker has some through lines, and the through line information is shown in the diagram in the common ancestors column. Here, these through lines suggest a specific pair of great-grandparents associated with some of the matches in this lowest cluster. If you can say how one person in a cluster is related to you, then you can suppose that everyone else in the cluster is related to you in a similar way. So all of the matches in this cluster are likely second cousins to the test taker through that same pair of great-grandparents. When you have unidentified clusters, those need more work. You can start by looking at the public trees for each match within the cluster. The type and size of each tree is shown on the left side of the diagram, along with a clickable link that will take you to that match in their tree. You might look for names or places that show up in several trees within the cluster. Those names or places might not yet be familiar to you. They are leads for further research. This approach is an excellent way to break through brick walls in your family tree. The left side of the diagram also displays other information that you have entered into your own Ancestry DNA results, including your notes and colored dot groups. The cluster diagram provides another way to view your research that you have already done. By the way, one common question is what is meant by the different shades of color in the diagram, such as the darker reds and lighter reds shown here. The short answer is not much. The shading is a minor aspect of the diagram as a whole. For most purposes, you can ignore the shading and focus on whether each cell is red, indicating a shared match, or white, indicating no match at all, or gray. We'll see some gray cells shortly. A detailed explanation about the shading is available in the website documentation in the Interpreting Clusters section. Before we move on, I'd like to show a second example, clusters over 90 centimorgans from another test taker. In some ways, this diagram is similar to the first. In some ways, it is different. The most obvious difference is that there are only 3 clusters here. 4 clusters is the ideal case. You only get 4 clusters if relatives from each grandparent actually are in the Ancestry DNA database. If the relatives haven't been tested, they won't show up in the clusters. Nothing to help with that except to be aware of it. The other obvious difference in this diagram is the strong vertical and horizontal lines that cross the entire diagram. You can see from the shared centimorgans column that this is a sibling of the test taker. It makes perfect sense that a sibling matches all relatives from all 4 of the test taker's grandparents. This example also shows how the cluster dot information from the Ancestry website is displayed in the cluster diagram. Test taker has started using the colored dots to indicate matches that they've identified in different parts of their tree. There's no surprise that those colored dots match well with the clusters. Once you understand your closest matches, you can generate clusters for more matches with lower centimorgan values. This time, let's generate a diagram that includes all matches over 20 centimorgans. I'll choose that radio button and slightly rename the file. And I'll click processed saved data. As before, the diagram will open in Excel. When you generate clusters under 50 centimorgans or so, you start including matches who share only 1 or 2 DNA segments with you. That changes the information in the clusters. Instead of representing grandparents at the 90 centimorgan level, the clusters at the 20 centimorgan level represent individual DNA segments. You can zoom out and you'll see that there's a lot more information in the 20 centimorgan diagram. Here, you can see an area with a gray background that links several clusters. There's a group of 1, 2, 3, 4 clusters within this larger area, including the gray background. The gray background to those clusters indicates that they represent individual DNA segments that came from a similar location on your family tree. They probably passed through several of the same most recent ancestors, branching out at possibly the 3rd, 4th, 5th grandparent level. You still need to do the research to identify each cluster, but for each cluster that you do identify, the gray background can also narrow down the choices for several other clusters nearby. As in the 90 centimorgan diagram, there are overlap areas in this 20 centimorgan diagram as well. Here is an area that includes some of the first cousins that we looked at earlier. Now there are many more matches on either side of the overlap areas. Here is a cluster that includes a couple of the second cousins that we looked at earlier. The cluster now includes other matches of 40, 30, even 20-something centimorgans. 30 centimorgan match is certainly not a second cousin, but seeing them in the same cluster suggests that the weaker match shares some of the same DNA segments with those second cousins. Their common ancestors are further back than a grandparent, but they may very likely be ancestors of that grandparent. Instead of having totally unknown matches, you now have matches that you have localized to at most 1/4 of your tree. In fact, here is an overlap area that includes some of the second cousins from the original diagram. The clusters on either side of this overlap area likely represent 3rd and 4th cousins related to the test taker through great-grandparents. In this way, the more clusters you identify, the more leads you have to continue your research. As one final example, I'd like to show what shared clustering does with endogamy. Endogamy makes shared match lists very difficult to analyze by hand. Ancestry is the only testing site where shared matches with endogamy can be analyzed at all. This doesn't work with all types of endogamy, but shared clustering may be the only application that can make even this much headway. This is the cluster diagram for a 100% Ashkenazi tester. These are my clusters. Compared to the clusters we looked at earlier, this is a mess. There are all sorts of red marks sprinkled throughout the diagram not connected to anything. The diagonal has blobs rather than squares. These are all hallmarks of endogamy in Ancestry DNA results. But even with the noise, you can still see some details. Zooming in and looking at the shared centimorgans column, you can easily see that the straight line marks near the top of the diagram are from my father. The dark area nearest him contains matches related to me through my father's father. There's another dark area near the center of the diagram. This one doesn't overlap the marks from my father. It represents the matches related to me through my mother's father. And that's all I can tell from here. The rest of the diagram is just noise. I'm one of the unlucky ones with very few close relatives tested from the other branches of my tree. I actually have found matches through all but one of my great-grandparents. It's just that most of them are below the 90 centimorgan cutoff for this diagram. Clustering is a great way to make sense of the data that you have, but it also helps a lot to have the right people tested in the first place. I hope that I've given you some ideas for how you can use the shared clustering application. Here again is the direct download link, We also have a user group on Facebook where you can share with and learn from other users of shared clustering. Good luck with your research.